Our next presenter is Lisa Benecchi. Lisa is a rising senior at Eckerd. She is majoring in molecular biology. She spent the summer in Vijay Ramesh's lab, and she is from Paris, France. And she has a very special guest here with her today. Her mom, all the way from Paris.
So, hi, like Sue said, I'm Lisa. I worked in Dr. Vijay Ramesh's lab this summer, and my project was to understand the role of apelin signaling in NF2 deficient meningioma and schwannoma cellular models. The Ramesh lab focuses on neurofibromatosis 2, or NF2, which is an autosomal dominant inherited disorder with an incidence of approximately 1 in 30,000 people. It is caused by mutations in the NF2 tumor suppressor gene discovered here at MGH in 1993 and is leading to the loss of the protein myelin. It is characterized by bilateral vestibular schwannomas arising from the Schwann cells and meningiomas arising from the arachnoid cells of the meninges covering. These tumors, unfortunately, also occur sporadically in the general population and do not respond to conventional chemotherapies. Therefore, non-invasive drug treatment is much needed. Previously, the Ramesh lab conducted an RNA sequencing, which showed increased expression of a ligand named apelin in NF2 deficient cell lines. So if you look at here on this column, you can see the ratio of apelin expression in NF2 null cell lines, arachnoid cells in particular, over arachnoid cells expressing NF2. And you can see that the expression of apelin is positive, meaning that its expression is increased in NF2 null cells, and similarly in Benman1 cells, which are grade 1 meningioma cells. So there's a little color change here. It's okay. So what is apelin? Well, apelin is an endogenous ligand for the G protein coupled APJ receptor at the surface of the cell, and its signaling is mainly involved in cell survival and proliferation, cell migration, and angiogenesis, characterized by the formation of blood vessels. Interestingly, schwannomas and meningiomas are highly vascularized tumors, and what these tumors do is that they hijack this pathway by secreting angiogenic factors such as apelin, stimulating blood vessels to grow into the tumor, providing it with oxygen and nutrients, leading to rapid tumor growth. Therefore, my project was to characterize apelin expression and pathway readouts in NF2 null cellular models. So it involved culturing models relevant for human NF2, such as 3 cell lines, 3 meningioma cell lines arising from the arachnoid layer that you can see right here. And those cell lines were Benman1 sporadic meningioma cells, shown right here, MN1 meningioma grade 1 cells, and KT21 malignant grade 3 meningioma cells. I also cultured Schwann cells where NF2 was deleted using the CRISPR genome editing tool. My project also involved performing quantitative qPCR to test the expression of apelin in NF2 null cell lines, as well as Western blotting to check apelin pathway readouts in NF2 null cells. Forgot to click. So the first step was to validate the RNA sequencing results, which confirm increased expression of apelin in NF2 null cells. So to test on the RNA level, I performed qPCRs, which involved culturing NF2 deficient cell lines, isolating the RNA to synthesize cDNA, and then adding the apelin primer, as well as the cybergreen dye mix, also containing DNTPs in DNA polymerase, to perform the qPCR, which happens in 3 steps. The first step is denaturation, which separates the DNA strands in order to allow for the primer to anneal and extend the strands while binding the fluorescent dye to finally form new DNA strands which are emitting fluorescence. And then the results are shown as sigmoidal curves that you can see right here with an exponential phase corresponding to when the fluorescence is detected. And this phase gives a cycle number. Therefore, the higher the gene expression, the sooner the fluorescence is detected and the lower the cycle number. So these 3 graphs below are representing my results of the qPCR in 3 different cell lines: isogenic arachnoid cells, meningioma cells, and isogenic Schwann cells. The y-axis characterizes the apelin expression, and the controls, which are the triangle shapes that you can see right here, are cells expressing NF2. So if you look at the isogenic Schwann cells graph, you can see that the cells that are not expressing NF2, so the NF2 null cells, have a higher expression of apelin compared to the cells that are expressing NF2. Secondly, in the meningioma cells, we use KT21, which is our malignant grade 3 meningioma, and MN1, which is the grade 1 meningioma, as our meningioma cellular models, which are right here on the graph. And then we compared them to arachnoid cells expressing NF2. And again, you can see a very significant increase of the apelin expression in those cell lines. Finally, when looking at the isogenic Schwann cells, we compared Schwann cells that are not expressing NF2 that you can see right here to Schwann cells expressing NF2. And once again, we can see a very significant of the apelin expression increase of the apelin expression in the NF2 null Schwann cells. Then, not only wanted to know if the ligand was expressed in the cells, but also the receptor. So I connected a piece of qPCR and I couldn't find any results in our cells. So we tested on the protein level, and by immunoblotting, you can see that the receptor is expressed in the meningioma models and the schwannoma models. But we couldn't see, there isn't any difference in the signal in the cell expression expressing NF2 and the cell not expressing NF2. So now we had a question, which was, can apelin stimulate downstream signaling pathways in meningioma cells? And here's a reminder of the pathway. And when apelin binds to its receptor, it mainly activates those three downstream pathways: STAT3, AKT, and ERK. Therefore, what we did is that we treated Benman1 cells, which again are grade 1 meningioma cells, with apelin, and we stimulated them at different times. So here, it's important to know that when those three pathways are phosphorylated, they are activated, which is why we compare their expression, phosphorylated expression level. And we use this using densitometry analysis with ImageJ. And you can see here that for phosphostat3, it is indeed activated after 5 and 10 minutes of stimulation with apelin. We couldn't see any changes in AKT as all of the values are around 1. However, for phospho-ERK, we could see also a dramatic increase after 5 minutes of stimulation, meaning that when apelin binds to the APJ receptor, it activates the ERK pathway and the STAT3 pathway. And then we wondered, if we add an inhibitor that prevents the binding of the ligand to its receptor, can it affect this pathway as well? And so what we did was that we treated the cells with MM54, which is the inhibitor for the APJ receptor. And we did it, and we co-treated the cells also with apelin. So on the left side here, these are the cells that were only treated with the inhibitor at different concentrations. And here on the right side are the cells that were co-treated with apelin and its receptor. And so we compared them. So the idea was that the cells that were co-treated with the receptor and stimulated with apelin should have the same expression values as the cells that were only treated with the receptor. So when you look for phosphostat3, here those cells did not have any inhibitor, they were just stimulated with apelin, and they have a 3.6 fold increase. And then we added the inhibitor at different concentration. You can see a dramatic decrease in apelin expression, meaning, and the values also are a bit similar to when they were just treated with the inhibitor, meaning that indeed MM54 inhibits this STAT3 pathway. For phospho-AKT, we just did the results, and we can see again, so when we compare it to the cells that were just stimulated with apelin, we can see a decrease in the apelin expression except for 2 micromolar concentration. And for phospho-ERK, however, we couldn't see any changes upon treatment with the MM54 inhibitor, suggesting that there is maybe a potential other receptor at the surface of the cell able to bind to apelin. Again, we will have to look further into this, but keep in mind that all of these are preliminary results, therefore we cannot come to any concrete conclusions. And so finally, the Ramesh lab previously discovered that upon NF2 loss, mTOR is upregulated. mTOR is a major regulator in the cells and is involved in cell proliferation. So what happens, in other words, when NF2 is lost, mTOR is constantly activated, causing the cells to proliferate. And so we wondered, what happens if we inhibit mTOR? Can this decrease apelin expression? And so what we did was that we treated our two grade 1 meningioma cell lines, Benman1 and MN1 cells, with 3 different mTOR inhibitors, rapamycin, AK128, and RM006. And then we compared them to our control, which were cells just treated with DMSO. And if you look at this graph, again, the y-axis also shows the apelin expression. And if you look at this graph, all of these cells that were treated with the inhibitor have a significantly decreased apelin expression, meaning that there is a potential feedback pathway mechanism within the cell where mTOR is involved in apelin transcription. In summary, apelin expression is increased in NF2 deficient arachnoid meningioma and Schwann cells. Apelin 13 is just the amount of amino acids that is in apelin. So apelin 13 stimulation activates both STAT3 and ERK signaling in meningioma cells. Apelin receptor APJ is expressed in NF2 deficient Schwann and meningioma cells, and treatment with its inhibitor downregulated phospho-STAT3. Finally, treatment with mTOR inhibitor decreased apelin expression, suggesting a potential feedback pathway mechanism within the cell. In the future, the Ramesh lab will try and work on understanding the regulation of the apelin APJ downstream signaling pathway upon NF2 loss, studying the role of APJ receptor and whether apelin is capable of binding to another receptor, and finally explore the role of apelin in NF2 tumor angiogenesis and the microenvironment. In the summer, I had the opportunity to learn how to perform human cell culture, drug treatments on human cells, RNA isolation, cDNA synthesis, qPCR, SDS phage, Western blotting, and finally densitometry analysis using ImageJ. I would like to thank Dr. Vijaya Ramesh for welcoming me to her lab this summer. And I would also like to thank Swarupa and Roberta for training me during this 10 weeks and for being very patient and always helpful. And also Sue, without whom this project would have not been possible. And Colleen for always being here for us. And the people who came here and did their summer seminar series. And finally, Dr. Michel, who allowed me to shadow one of his surgeries. Any questions?
Any questions for Lisa? But you guys gotta help me out because I don't have them.
It's okay. It means I was clear.
That's okay.